Antarctica sits at the bottom of the world, a frozen land buried in ice for millions of years. Deep under this ice lies Lake Vostok, a freshwater lake about the size of Lake Ontario. Sealed off from the outer world for millennia, the lake is freezing, lightless, and subjected to crushing pressure nearly 400 times atmospheric pressure. In 1998, a joint U.S., Russian, and French team drilled an ice core down almost to the lake. The conditions at Lake Vostok are so extreme that it would seem nothing could live there, yet in the ice that had frozen off the lake, scientists found evidence of microbial life.
Lake Vostok is such a unique environment because it's buried under more than 3 kilometers of ice in Antarctica, and there is no light. There is apparently no food that we can think about, and there is a lot of oxygen, which is toxic for many life forms out there, and still there are microbes living there, and we want to know how they live there.
To answer this question, one of the DOE JGI's 2009 Community Sequencing Program projects will sequence the genomes of this microbial community. The Department of Energy is keen to learn more about these bugs, because these microbes must get their food by removing carbon dioxide from the atmosphere and fixing it into organic molecules. Since excess carbon dioxide in the atmosphere contributes to global warming, anything that could remove carbon dioxide is of great interest.
First of all, we are interested in the ways they fix carbon because they must do it very, very efficiently. They are not limited with carbon. There is enough CO2 there, but they're very limited with energy. So whatever they do, they must do very efficient. Also, there is a very high oxygen pressure, and oxygen is known to interfere with carbon fixation. And these guys managed to fix carbon with very small energy and with very high oxygen pressure.
Although land plants also remove carbon dioxide from the atmosphere, the process is inefficient and energy intensive. Lake Vostok microbes, by contrast, could be the most efficient carbon fixers on Earth. But studying these microbes will involve several technical challenges, and will require the efforts of a collaboration that includes not only JGI researchers, but also Craig Carey of the University of Delaware, and the project leader, Brian Lanoil, formerly at UC Riverside and now at the University of Alberta. Lanoil's group at UC Riverside will process the ice core and extract the bacteria. Extraction is a very challenging procedure because the scientists must be careful not to contaminate the sample with even one bacterium from our environment.
Our collaborators in UC Riverside got this ice, and they're going to do the procedure of decontaminating the ice core and getting the microbes out of there, which is extremely complicated, and I wouldn't do it myself, but they are experts. And we get the microbes, we put it in our sequencing facility, and then we get their genes.
To analyze the genomes, the DOE JGI will make use of the techniques of metagenomics.
Metagenomics, quite simply, is the application of high-throughput sequencing to DNA extracted from environmental samples. Now, why I would be interested in this necessarily, it's because most organisms that are present in environmental samples can't be grown on an agar plate. So we haven't had access to the genomes of most organisms, maybe 99% plus of the organisms in the environment. So this is a very quick way to get genetic information, metabolic pathways and the like, from organisms that can't be grown in culture. Plus, you also have the added benefit that you can look at complementary metabolism between different members of the same cohabiting members of the same community.
The genomes will be deciphered using the DOE JGI's unique integrated microbial database for metagenome samples. In addition to discovering novel carbon fixation pathways, the researchers hope to find other unusual adaptations to the extreme dark, cold, and pressure of Lake Vostok. Because Lake Vostok's conditions resemble the environment found on some other planets, like Jupiter's moon Europa, the genomes of the Vostok microbes could even yield clues to the nature of extraterrestrial life. They might also provide a window into the evolutionary past of our planet, preserved pristine in life under ice.